Okay, we're going to start part 2 of chapter 7. We're still in section 7.1. That was a really large section, so we just have this last part to complete, and then we'll start in section 7.2. So the last part of this, we've talked about nutrients, where they are, what they need. Now we're on this part of the absorption of the nutrients. We know that to get in and out of the cell, we've got to transport through the plasma membrane. Right, and a lot of these microbes have a cell wall, like bacteria and the fungi. So we're going to talk a little bit about those challenges. So our cell membrane, you're going to hear that it's selective or differential. That simply means that not just anything can cross the cell membrane. Only certain things can move in and out of the cell. And it's basic biochemistry. It's size, it's charge interaction. Okay, and the cell wall, by the way, it is not selective. It's non-selective, which means different things can come in and out, okay, without a lot of requirements. So it's really the cell membrane that is controlling everything. So before we get into how we're going to absorb these materials, we got to review diffusion and osmosis. So as a reminder, diffusion is simply the movement of molecules from an area of high concentration to lower concentration. We tend to think about this in terms of across the cell membrane because in biology and microbiology, that's what we're talking about. You had a high concentration of glucose outside the cell. It can diffuse across the membrane into the cell where it's a lower concentration. But I just want you to keep in mind that diffusion doesn't have to happen across the cell membrane. Diffusion happens every day. If you were cooking dinner or if you light a candle in your house, those scent molecules diffuse from the area where there's a high concentration, wherever you have your candle or your dinner cooking, throughout your home, where it was a lower concentration. Okay, so it's just a movement from high to low concentration, and it is a natural movement. When microbiology and biology, we're usually talking about substances moving across the membrane, because we're talking about cells. And we say concentration gradient, that simply means we have more molecules on one side than the other. Right? And we'll talk about permeability at some point, I believe. Okay, so why do things diffuse? This is something important to mention. Brownian movement, or sometimes called Brownian motion, this is just the random movement of a molecule or a substance, an element, whatever it may be, in a gas or a liquid. It never stops moving. Nothing stops moving. All molecules and substances just have their own random energy of movement. Okay, so and here's an example saying if this is a sugar cube. I don't know who actually ever uses sugar cubes, but let's just pretend you put a sugar cube into your coffee and it's going to break down. This is a high concentration. It will diffuse and pull apart and you will have sugar throughout your coffee. So we're going to talk about that just for a minute. If you did nothing, if you just put some sugar in your coffee, it's going to dissolve and then diffuse throughout your drink. Okay, you could obviously speed that up. Some of the things that speed up diffusion would be, of course, heat. Heat is that motion of the kinetic energy of molecules, right? It's movement. So if you have more heat, you have more movement of all the molecules. You're going to speed up diffusion. Concentration gradients. If you have a lot of whatever molecule on one side of your cell membrane and not as much on the other, it turns out we're going to have more collisions and we're just going to have a faster process of those molecules colliding and moving to the other side. Also, another thing, this isn't mentioned on here, but if you stirred it, you would move and you would increase diffusion. So that's just some things to think about. Now, we're going to think about it, of course, in microbes. A term that's really important for you to understand is called net movement. So a lot of students don't really know what that means. If we say the net movement of the sugar molecules was to the top of the coffee cup, that means the general movement of the molecules. So this was the high concentration down here at the bottom. This is all low concentration. It's generally, they're all moving this way. Does that mean that every sugar molecule goes straight up to the top? No, they're going to bounce and hit the side, then maybe hit this side, then hit over here, then hit another molecule of coffee. So they're making these collisions and bouncing around all different directions. But what is the net movement? What is the general movement of all the molecules? This direction, okay, or just away from this source. That's what that means. Okay, and we're going to review that when we get into osmosis because that's going to be important that you understand that. Okay, so here's another way to think about this. Once we have our sugar all diffused through here, and you have it evenly spaced throughout, they're all still moving. They never stop moving. There is no more net movement. They're not all moving in one general direction anymore, because what they find is they're all spaced throughout, so they're all moving in every direction. There would be no net movement, but it doesn't mean they stop moving. So osmosis, we said diffusion. So when we say diffusion, we're talking about solutes, the diffusion of a solute. Osmosis is the diffusion of water. We usually refer to this across a selectively permeable membrane. Because the water is moving from one location to another, it's usually through a membrane. Okay, so again, here we are saying cell membranes are selective. Only certain molecules can pass through, and it depends on the cell what gets in and out. And this is an important thing to mention. No living things, whether we're talking about us or a microbe, we don't have an ability to actively push water in any kind of location. And that's important for those of us that are multicellular and have these big compartments. We can't move water on cue. Okay, we can't push it around. So the only way we can move water is by actively pushing solutes around. And that's where we say this amount of water that enters and exits a cell can really have a lot of effects on our cell activities and survival. So osmosis is how water is just naturally moving. And it's still moving, by the way, from high to low concentration. But we're talking specifically of water molecules. Okay, so the concentration of sodium chloride is not going to affect the concentration of the water. Okay, so let's kind of look at this. Here's some examples. And this is going to be kind of our cell right here in this cup. And we're going to put the membrane around it. We're going to say it is selective in that these molecules in here, which we'll say, let's make it glucose. And the red little dots will be glucose. They're too big to get out of this selective permeable membrane, okay, our cell membrane here. And we're in a glass of water with only water molecules, which are denoted by these blue dots. So if we're just looking at this, and we know that our solute can't diffuse, okay, it can't move across this membrane. If it was able to, it would move out here. It would go from high to low concentration, right? It can't do that, but water can move. So you have to be looking at where is there more water molecules. There's more out here versus in here. So water moves down its own gradient. And we say moving down, think of like on the top of a hill. You're going from a high location to low. We're going high to low concentration. Water will flow in this direction because this is where there's less water. That's the natural movement of water. So here you're seeing more water come in, and now some of that solution is getting pushed up into this tube and pushed up into this tube. Water will just keep coming in here, okay, because there's solutes in here and not out here. If we wanted to reach an equilibrium, we would have to make the pores, sorry, I lost my mouse, there it is, the pores on our membrane bigger so these solutes could come out. And then the solutes would be equal on the outside versus the inside, and then the water would equalize out. Okay, so osmosis is that movement of water from high to low concentration. So we can generally say where there's a lot of solutes and the solutes can't get out, that's a low water concentration. Water flows in. Okay, so let's keep going with this. Okay, what drives osmosis? This is what I'm saying. Solutes do. If we have these two cups, let's say we have two cups here, the exact same volume, but when we look at this one, most of the space is taken up by our solutes, which means there's less water molecules here than in here. Hopefully that's making sense to you. So here we're going to say this is a lower water concentration. It's high concentration of solutes, lower water concentration. This one has higher water concentration, lower solute concentration. So now let's talk about diffusion. Diffusion refers specifically to the solutes. Which way would these solutes diffuse? If we let these two, there we go, if we put these together and let these touch and made it so that the solutes could go back and forth, would say that the pores are big enough to let these solutes diffuse, which direction are these solutes going to diffuse? From high to low concentration. So these solutes right here are going to diffuse to this side. Some of these are going to come over to this side, right? They're just bouncing around, bouncing. Some are bouncing here. But eventually, the net movement, there's that word net movement again, is going to be from this side over here to this side. Okay, we're moving to the right side until you have the equal number of solutes on either side. And they're always going to keep moving back and forth and back and forth. They don't stop once the concentration equalizes. Now let's do that same experiment, but let's say that these solutes can't move. Okay, in that case, if we put these together and we let only water diffuse, we would say osmosis, which way is the water going to go? Well, which side has more water molecules? This one does. Okay, anywhere there's blue, that's water. So there's more water here than here. So water diffuses that way. Solutes would go this way, water would go that way. Okay, so diffusion of solutes is going to go the opposite way of the water. Okay. And what you have to be considering, can they both move at the same time? Can only the solutes move? Can water move, but not the solutes? Okay, because that's going to affect whether you're having diffusion or osmosis or both. Now, taking all that and applying it to our microbes. When we use these words, and in your textbook, they use the word condition. We're talking about the environment the cell is in. Okay, so if we say an isotonic condition, we mean an isotonic environment. Iso is neutral. So what it's referring to is solute concentration. We're going to talk about isotonic, hypertonic, and hypotonic conditions. Those words, the iso, the hyper, the hypo, are referring to solute concentration. As a review from chemistry, remember these brackets mean, or they indicate concentration. So this is external environment, the solute concentration, equals the internal cell's solute concentration. So, and I'm going to just throw a random number out there. If I had 100 solutes on the outside of the cell dissolved in water, I would have 100 solutes on the inside of the cell dissolved in water. It would be whatever it is, or whatever the molarity is. Let's not use number. We'll say 3 molar versus 3 molar. Okay, so the concentration is the same. In that case, you're seeing arrows in both of these examples. This would be, say, a bacteria. It has a cell wall. Water flows in and water flows out. Water flows in, water flows out. Okay, this would be a cell without a cell wall. Let's say this is an amoeba. Okay, so we got water flowing into the cell and water flowing out. Notice when we talk about net movement of water, which way is the net movement? There is none, because what this is showing you is that there's concentration of solutes are equal on either side of our cell, on the inside versus the outside. So water flows in at the same rate it moves out. So when we look at net movement, right, it's the same going in versus out. So there is no net movement of water in this case. And here, it doesn't matter if your cell had a cell wall or if it didn't. This would be the human body. We have isotonic conditions around our cells, in our cells. Okay, everything is isotonic. Okay. And this is the most stable environment for cells, keeping in what we would say a steady state. Right, now let's see what happens when you're not in a steady state. What if you're in a hypotonic environment? And here I'm underlining conditions. It gets important that you think about this, because if I'm saying that the environment around the cell is hypotonic, remember that hypo is referring to the number of solutes. So what I'm saying is there is hypo means low. So if you heard of someone with hypothyroidism, they have low thyroid hormone. Hypo is always low. So if you have a low number of solutes outside your cell, that means you have more water. Right? Okay, remember what that means. So hypotonic, there's less solutes out here around your cell, which means there's more water. Now we have to say that in comparison to a second solution. So there's one solution out here, which is hypotonic. We're saying that in reference, in comparison to a second solution, which is that inside of our cell. Right? If this is hypo, we're saying this is hypotonic in comparison to this solution, which is the word hypertonic. Hyper meaning many. In that case, we're saying the outside conditions are hypo. Water is flowing into our cells. Why? Less solutes means more water. This is higher solutes, less water. So if the solutes can't move, that means water is going to flow into this cell. Okay, so far so good? There's a problem with this. And for example, what would be a hypotonic condition? This would be, think about like living in pond water. Okay, pond water does have dissolved substances, but it's more low solutes than the organisms living in there. If you're a bacteria and you're in a bacteria up here with a cell wall, and you're living in a hypotonic environment, water net flow, you're seeing the arrows now, comes into the cell. However, eventually only so much water can come in because of something called turgor pressure. The cell wall out here in pink is very strong, and it prevents that cell from exploding or bursting. Even though water flows in, only so much water flows in that it fills up. Sorry, I keep losing my mouth. And this yellow plasma membrane is gonna push against our pink cell wall until it's firm or turgid, and then it stops. You can only put so much water in there, and then it stops due to the pressure from the cell wall. This is a problem, however, if you don't have a cell wall. Okay, if you're an amoeba, if you're in a paramecium, and you've got all this water flowing in, your cell is going to burst. There's nothing preventing that. The water will just flow in, flow in, flow in until this cell explodes. Okay, so that is a bad thing. You would have to find a way to overcome that, okay, so that you don't explode if you're a single cellular organism living in pond water. What if you live in the ocean? That's hypertonic. The ocean has many, many solutes dissolved in it. More solutes on the outside than the creatures living in the water. And so hypertonic, remember, hyper is describing the solute number. We're saying hypertonic conditions. That means the environment is hypertonic in comparison or in relation to the inside of the cell, which would be hypo. If you say iso, we mean, again, we're always comparing two solutions, watery solutions, right? And we're always looking at solutes. If it's iso, they're the same. If I say the environmental conditions are hyper, that means the inside of the cell is hypo. If I say environmental conditions are hypotonic, the inside of the cell is hyper. So I'm saying it in relation to something. You can't just say, oh, well, the ocean is hypertonic. Well, in relation to what? Okay, so you need to have a comparison. It has to be more solutes versus something else. In our case, we mean with organisms. So remember, where there's more solutes, there's less water. Which means the water, there's a lot of water in here and less solutes. Where's the water going to flow? From high to low concentration. And so if all the water flows out of this little bacteria, something called plasmolysis occurs, which means the cell membrane pulls away from the cell wall and this little guy will shrivel up and die. If this is our amoeba, all the water, you're seeing the arrows come out, that means the net movement is in one direction. It's all the water is leaving because there's more water here than here, the concentration. Okay, we're looking at solute concentration. We have way more solutes out here than here, which means water is less on the outside versus the inside. So when all the water goes out, the cell shrinks up and becomes distorted and it will die. So this can limit the growth of our microbes. If you think about, we use salt as a preservative, and there's sugar solutions that preserve food, like honey can be around for thousands of years and it's not going to spoil. Why is that? Because many organisms can't survive in a hypertonic solution. What will happen? All the water will leave and they're going to shrivel up and die. So that's a very good preservative. And something to introduce you to, because you're going to hear about osmotic pressure, or sometimes it's called osmotic potential. So I want to make sure you're clear on what that is and don't be overwhelmed with this. A very easy way to think about it, I'm just simplifying this for you. This is not chemistry, but if you had a solution like this picture over here that has a high concentration of solutes, we say that it has high osmotic pressure, or you might say it has high osmotic potential, because it has the potential to draw water in, right? Because there's not as many water molecules here, so it has the potential to pull water in. If you have a low solute concentration, you would have low osmotic pressure or low potential. Okay, it's not likely that water is going to flow this direction. Water is probably going to flow out. So what do you do if you live in one of these environments. We said if you're in isotonic conditions, you're not very stressed out because your water is not leaving you and you're not shriveling up, or water is not coming in and you're not exploding. So any bacteria that live in our bodies are probably happy in their isotonic conditions. If you're a bacteria or amoeba living in fresh water, okay, we said, okay, if you're a bacteria, water's going to flow in, because this is hypotonic on the outside, hypertonic on the inside. Water flows into a hypertonic solution because that's less water. And water flows from high to low concentration. So when that water flows in, our bacteria are okay because they have the cell wall. They don't really have to do anything special. They can survive. They're fine. If you're an amoeba, you're going to explode. You don't have a cell wall. So in that case, they have these structures called contractile vacuoles. Well, literally, it's like a little baggie or sack that fills up with water, and they just shoot it out. And it takes energy to do this, but some of them use it for motion. Okay, so it fills up, they shoot it out. It fills up, they shoot out. They're just constantly squirting the water out. If it's a hypertonic environment, you must find a way to restrict the water from leaving the body. And there's different ways that organisms can do this, but a lot of organisms just don't live in hypertonic environments. There's one cool one I want to tell you about, the halobacteria, which are the ones that live in very salty solutions like the Great Salt Lake. They actually absorb salt, and then they make their cells isotonic with their environment. Okay, so they're actually able to process that and not become have toxicity from it. So only certain bacteria can do that. Okay, so this was all for our diffusion of how these cells are getting the molecules in and out. Okay, and then we talked about osmosis, how they're getting water in and out. So another part of this is something called facilitated diffusion. So we're saying diffusion, so we're talking about our molecules. And essentially this means when anything diffuses through a protein, a transport protein. So that word facilitate, if you facilitate something, you might help. Okay, think of it like a helping it. So this is what we would say, a mediated transport. So you're going to move your whatever molecule's diffusing through a protein. There's two types of these proteins. Carrier proteins, which you see here, or channel protein. If it's a carrier, it does what it sounds like. It actually attaches to the molecule. It's specific. Okay, it changes shape. That's what this word conformation means. It changes its conformation or shape, and then deposits that material on the other side. So if it was on the inside, they can carry it to the outside. If it's on the outside, they can bring it in. If it's a channel protein, this is much simpler. It's just a channel. Think of it like a hallway. You just open it up. Whatever substance it is will flow in through diffusion. Okay, it's just concentration based. And here's a picture to show you this. So here's just diffusion. If you just diffuse across a membrane, you're going from high to low concentration. This would be facilitated diffusion. Okay, when you're going through an actual protein. This would be a channel. It's literally just like a pore in the membrane. It is specific. Like we have ion channels. Your sodium channel is not the same thing as a potassium channel. Like potassium can't go through the sodium channel. So there is some specificity there. This would be a carrier protein here. Here's your substance. It's going to attach to your carrier protein, and then it's going to deposit it on the other side. I don't know why I changed shapes. But all of these are types of diffusion, okay, which we would say is passive transport. It means we didn't have to use any energy to do this. Okay? Our molecules are just going to diffuse based on their natural motion of diffusion. That's natural. We don't have to do anything. That's the complete opposite of active transport. Okay, so our active transport is when the verbiage for this, we say against a concentration gradient, which essentially means we're going from a low concentration to high concentration. Think of it again like you're on a hill. This time you're at the bottom of the hill. You're going against the gradient. That means you got to go up the hill. You're at a low position. Now you're going to go to a high position. So low concentration, it takes work to get to the top of the hill, right? You don't have anything helping you. You've got to use energy to get to the top of the hill. So if you're going from a low concentration to a high, then you need to exert and expend your energy. You're going to need ATP. And that's what this picture is showing you. This has to go through some kind of transport protein. Okay? It can't just diffuse naturally through with active transport, or you need something to push it through because it's not a natural movement. If there's already a high concentration in here, it would favor diffusing that way. When we're going low to high, that means you're going to have to pick it up and push it to the other side, and you're going to need energy to do that. These transport proteins are often called pumps. So you can also use that to your advantage to think of like pumping up a tire or something. You would be using energy to do that. So this is an active feature of a cell. Another active transport would be something called endocytosis. Cytosis is within the cell. Endo is within. So this is bringing into the cell. Literally, that's what this means. There's different ways that we talk about this, but the two main types of endocytosis are phagocytosis and pinocytosis. Phagocytosis is cellular eating. Phag means to eat. So this is literally cellular eating. Pino is liquid. So this is cellular drinking. If they're going to use their pseudopodia, which are their little feet that come out, called fake feet, they can envelope a particle or a solid material. They can wrap around and bring it in and put it in a vacuole and eat it. If it's a liquid, they can kind of enclose it and bring in a drop of liquid. There could be something dissolved in the liquid. It's the same idea. They just sort of make an enclosure around the liquid, make a vesicle, and bring it inside. This does require energy to do this, so this is active. Okay, let's check our time here. Oh boy. Okay, we're going to stop on that one. And we'll come back.